Okay, so now we talk about splicing and alternative splicing. Splicing is essentially a process of removing introns and joining exons together. So now we need to know what are exons and introns. So exons, these are information that is coded. So coding information or important or real information, right? Let's just say this is the actual information we need. Let's not say coding because introns are also coded. So that's important to know. Both are coded. This is the important information. The real information. The significant information. The actual information. You can go on and on with the synonyms. But introns are gibberish. Not important. See? You get it now? So see? Intron sequence that does not code for a protein. But it does code, right? But it will enter the code. It will enter the transcript. But it won't be translated. It will be transcribed, but it cannot be translated into a protein. Okay? So now how do we splice? What is the mechanism? So in order to know the mechanism, we need to know the things that use it, the SNRPs. So SNRPs are uracil-rich proteins, uracil-rich RNA or proteins. Actually, they are RNA binding proteins. And they form base pairs with consensus sequences at the ends of introns. Now let's see how does that happen. Right here in blue, this is the exon 1, this is exon 2, and orange is the intron. This site is called as the splice donor site, and this site is the splice acceptor site. I'll tell you what that means a bit in a while. These are U1, U2, U3, U4, U5 proteins. They are gonna come here and get attached to the intron. Let's see how does that happen. We have a SNRP and ATP coming in. The primary transcript, you see the primary transcript right here. It is gonna combine, the whole of it is gonna combine to SNRP and form a spliceosome, right? So spliceosome is the primary transcript, SNRP proteins, and these uracil-rich proteins. These are all SNRPs, basically. And they're gonna get attached. Now what is gonna happen is that this OH at the A site, this is a splice site, the intron, OH is gonna attack the P of the 5' end of the splice. Sorry, of the intron. Because it attacks here, you see in the next diagram, it forms a lariat structure. This is a lariat structure. See, if you have seen Western movies, you would see, those cowboys, throwing ropes in a loop-like manner. That's a lariat, actually. So that's what it's called, lariat. When this kind of attack happens, what is the next attack? So the next attack is that the splice acceptor site. So this is the splice donor site. This is a splice donor site. This splice donor site is going to attack the splice acceptor site. So this splice is going to get accepted here. So it is donating to this acceptor. So the OH here at guanine is going to attack here at the P site. And when these two exons join, the whole lariat of the intron right here, it is going to get removed. And that is how splicing is done. Right? That is it. Pretty easy. Next, we have the alternate splicing, a very easy example, a very simple concept. We don't need to do quite details of this concept. Is that we have exon 1, exon 2, and exon 3. We can combine it in different ways. We can splice exon 3 and 2 together. We can splice exon 1 and 2 together and remove 3. We can splice 1 and 3 and remove 2. List goes on. Some example is a tropomycin. We can form different isoforms of tropomycin with alternate splicing. What does it mean? That we have one essentially long mRNA right here for tropomyosin, but what we do is that we create splices and we alternately join splices to form various different isoforms of the tropomyosin. And that is it for splicing. Thank you so much.